Okay, hi everyone, and welcome again to Biology 2. So this time around for Module 1, this is the third video which we're going to be talking about monohybrid crosses. So this is very important, a skill that you will be needing, especially as you go to take your college entrance examinations. There will be a number of this kind of questions. So it would be very, very important for you to really master this how to answer monohybrid crosses, and then on the next video, dihybrid crosses. So first of all, when we talk about monohybrid crosses, you already know that we're just talking about one single trait. We're talking about just one gene pair. So for example, we're talking about height. So you talk about here the allele for tall or allele for short, or probably you're talking about only the character for color of flowers. So probably you might be talking about the red and the white flowers only. So it's simply stated, monohybrid crosses are crosses between two parents wherein you are just taking note of only one single trait. So for example, as Mendel has already done, crossing two pea plants that differ in stem size, one tall and one short, and the big T, which is the dominant allele, will be for tall, and the small t, which is the allele for the recessive, will be the dwarf one. And of course, you're very familiar that there will be three possible combinations here, and that will be a big T, homozygous dominant. So this will be a tall plant, homozygous tall plant. And then you have your heterozygous condition, wherein it's the big T and small t. So since the big T is the dominant, therefore this will also be a tall plant. And of course, you already know that the homozygous recessive, the small t and the small t, will be a dwarf plant. So let us suppose that the parents, the P, would be a cross between a homozygous dominant big T and a homozygous recessive small t. So in this case, the question would be, how are we going to get the F1 generation? So this is quite simple. You already know this, very familiar. So in this case, the tall, which is homozygous big T, and the dwarf homozygous small t, first of all, you're going to be using the Punnett square here. It's very familiar as well. So in the Punnett square, what you're going to be doing is you'll be predicting the genotypes and the phenotypes of the offspring by the use of splitting what we call as the gametes. So the first step in order to do the Punnett square is for you to determine the gametes, and this is done by splitting the genotypes of each parent. So if this is your cross, the homozygous dominant and the homozygous recessive, what you do is you get the gametes simply by splitting the letters, simply by splitting the 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 genotype of your particular parent. So in this case, the homozygous dominant will be gametes producing or gametes containing the dominant allele, the big T and the big T, and the other parent will be contain or splitting it into the small t and the small t gametes. So that's the first step that you need to do. Then the next step is for you to make use of these gametes and then put them into the Punnett square, wherein the gametes of the first parent you will line on the right side, and the gametes of the second parent you will be lining on the top side. And it's simply a matter of combining the gametes together in the planes of the Punnett square. So it's quite easy actually to do this. So you have here a big T and small t, you combine the next one, it becomes big T and small t, and the next one you combine it big T and small t, and also the other one is big T and small t. So in this case, you will now be noticing that this will be containing tall, the other one is tall, another one is tall, and another one is tall. So the phenotypes of all we're gonna talk about what would be the genotype, what would be the phenotype, the genotypic ratio, and the phenotypic ratio. So based on the Punnett square, you already know that the big T and small t would be 100%. The phenotypes would be tall plants, 100%, as you can see here. And the genotypic ratio is, as you can see, there are none of the homozygous dominant, 4 of the heterozygote, and none of the homozygote recessive. So simply reducing the numbers, you will get 0 is to 1 is to 0. And further reducing this, we remove all of the zeros. The answer for the genotypic ratio is 1 heterozygote condition, or 1 big T, small t. When we talk about the phenotypic ratio, you just look at the phenotype. So since we all know that all of them are tall, therefore there are 4 talls and there is no short. Reducing the number, it becomes 1 tall and no short, and just simply reducing again, removing all the zeros, it becomes 1 tall. So that is how you will be getting the genotypic ratio and the phenotypic ratio as well. Okay, so in this case, there's another way to do that, which makes it easier. So since there are two gametes produced where it contains the dominant allele, and there are two gametes produced where it contains the recessive allele. So you can just write two T's and then another two small t here. And then when you combine them together, you can have two t combined with the two small t's, and then you will have four big T, small t. So the big T over here is here, the small t is over here, so combine them together, and then you multiply the numbers. 2 times 2 is equal to 4, and actually that's the same thing that we got a while ago here in the Punnett square. So we have 4 of those big T, small t, or the heterozygote condition. So this can be a shortcut that can be done to make it a bit easier. Okay, so that's the F1 generation. Now, let's just continue on. How about if we get the next part? So we already have the parentals and we got the F1 generation. Next step is for us to get the F2 generation. So if we're going to get the F2 generation, in this case, you get one F1 generation, which is the heterozygote, and then cross it with another F1 generation, another heterozygote. So the question would be what kind of uh, progenies we will be getting there. Of course, you have to take note of the genotype, the phenotype, and also the genotypic and the phenotypic ratios. So you're very familiar with this already, so let's just go straight to it. First of all, determine the gametes produced by each of the parent by splitting. So you already know if the cross is the heterozygote by heterozygote, the gametes would be first one is the big T in one gamete and the small t in the other gamete. And that will be the same case with the second parent because they're the same. It will also contain the big T and the small t as their gametes. Next step is for you to make the Punnett square, wherein you will be putting the gametes on one side and the other parent's gametes on the top side. Combining them together, you will get the genotypes and the phenotypes of this cross. So you already know that combining the first will give you homozygous dominant big T. Combining the second pair, you get a heterozygote big T small t. And then combining the third pair, you will get a big T small t. And the last one, you get a small t. So in this case, let's talk about the genotypes. So in this case, you will have here the following. Genotypes would be one should be one big T and big T. This will be the dominant. The next naman is you see here that there are two big T, small t, which is the heterozygote. And the last one is that you will get a one small t, small t. So phenotypes, you just look at it. So since this is dominant, it's tall. Since this is dominant, it's tall. Since it's dominant, it's tall. And then since it's both recessive, it will become a dwarf. So the phenotype will be three talls and then one dwarf. Talking about the genotypic ratio, then you will have one homozygote dominant is to 2, heterozygote is to 1, homozygote recessive. Now, please take note, one of the things you have to take note is it's automatic when if you have a heterozygote cross with another heterozygote, and we're talking only about monohybrid crosses, then you will always have a genotypic ratio of 1 is to 2 is to 1. One homozygote dominant, two heterozygotes, and one homozygote recessive. And that will be an automatic genotypic ratio for heterozygote cross with a heterozygote. In terms of the phenotypic ratio, you will also have an automatic ratio. And in this case, you will have how many? 1, 2, 3 talls and 1 dwarf. So it will be a 3 is to 1 ratio. So any kind of heterozygote cross with a heterozygote, you will always have a genotypic ratio of 1 is to 2 is to 1 and a phenotypic ratio that is 3 is to 1 ratio. So that's very easy for us to take note. So you can also do the fork method, and this will be quite faster compared with the Punnett square. So if your parentals is a homozygote dominant and heterozygote, and you cross them together, then combining the first with the first, you will get a big T and small t. Combine the first with the second, also a big T and small t. Combining the second with the first letter of the second parent, big T, small t. And the last second and the second letter, big T and small t. So you all have all 100% heterozygote, and all of them will be tall as well. Now, to get the F2 generation, you just get two of this F1 generation, and then you just simply cross them together using the fork method. So this will be tall, this will be tall, and then all of them are tall. So you just could get one. The first combined with the first letter, you get a big T and a big T. The first combined with the second letter, the second parent over here is supposed to be here. You get a big T and small t. And then the second letter of the first parent combined with the first, that should be the first, big T and small t. And finally, the second, and the second will be small t and small t. So using the fork method, now you will be able to get that it is 1 is to 2 is to 1 ratio. So it's quite easy if you're going to be making the fork method. You just have to make sure that the letters are combining are correct. Okay. Now there are other type of crosses. How about if you would be you already have done this homozygous already, so it's right done. You just combine them. So you just only need one box for this one because it's the same as we have shown a while ago. Okay, and how about if you have a heterozygous combined with the homozygous? So in this case, you just again get the gametes. There's still a shortcut here. So you put the big T over here and the small t on the other one. And then you put, since there's only one kind of gamete, you just put one small t over here. And then you combine them together. You will get a big T, small t, heterozygote, and a small t, which is the homozygote recessive. So if you talk about the genotypes, you will get 0 for the big T, the homozygote dominant, because there's none of this. 1 for the heterozygote, and 1 for the homozygote recessive. So their genotypic ratio is 1 big T small t is to 1 small t small t. Now this is what is interesting as well. This is also an automatic ratio, because if you're going to be crossing together a heterozygote with a homozygote, whether it's a homozygote recessive or at least for this case homozygote recessive, you will always have a 1 is to 1 ratio for this one, as you can see here. And the phenotypic ratio or the phenotype will be 1 tall and 1 dwarf, and the phenotypic ratio will be 1 tall and 1 dwarf as well. Okay, so that's quite easy. So let's look at some examples here. Cystic fibrosis is an autosomal recessive disease that is lethal, affecting Caucasians. It affects transport in tissues, causing mucus to accumulate in the lungs and cause infection. So it's quite a dangerous disease. If two parents are heterozygous for cystic fibrosis, what percent of the children is expected to have the disease? Okay, so for this question, of course, the first thing you're going to be doing is to determine what are the symbols, which is the dominant, which is the recessive. So in this case, we already know from the problem that it is a recessive disease. So the cystic fibrosis is controlled by a recessive gene or recessive allele. So it will be symbolized by small letters. We also know that since this is recessive, then the other character, one that does not contain or does not produce cystic fibrosis, will be the dominant allele. And that is what we call as a normal condition. So in this case, you have three possible combinations. So you have the capital C, which is the normal. So we already determined that the big C will be the dominant, and that will be the normal condition. We already determined that the small c is the recessive condition, which is the cystic fibrosis gene. So you have three possible combination here. The big C and the big C will be normal. The small c and the small c will have cystic fibrosis. And the heterozygote condition, the big C and the small c, will be a carrier. But since it contains a dominant allele, then it is normal. But it is considered to be a carrier of disease because of the presence of the recessive in the heterozygote condition. Now, if both of the parents would be heterozygote, therefore the father will be a big C and a small c, and the mother will be a big C and small c. So what kind of gametes they will be producing? It will be like this. One would be big C, the other will be small c, and the egg cells produced by the mom would be containing one big C and the other one would be small c. Okay, so that's how you get the gametes. Now, putting them together in a Punnett square, so the big C and small c from the father is over here, the big C and the small c from the mother is over here. So you combine them together, you have a homozygote dominant on the first pane, you have a heterozygote on the second pane, you have a heterozygote again on the second pane, and then you have the homozygote recessive on the last pane. So the genotypes would be one homozygote dominant, two heterozygotes, and one homozygote recessive. For the phenotypes, this would be containing the normal, This will be normal but carrier. This is normal but carrier, while the last pane will be one with cystic fibrosis. So there are three normals, and there will be one with cystic fibrosis. I just forgot to write it here. So what's the genotypic ratio? The genotypic ratio would be one big C, two big C, small c, and one small c. While the other, the phenotypic ratio would be, it's not tall, but rather it should be three normals and then uh, one cystic fibrosis. So that's how you are able to get the answer for that one. To use the fork method, you can just simply combine the first and the second letter from the second parent, the second letter from the first parent with the first letter of the second parent, and then you get here the last combination. So it's very simple to get the answer. So you have here normal, cystic fibrosis, and the other one is normal. So you would have there the 1:2:1 genotypic ratio, and then you will have three normals is to one cystic fibrosis phenotypic ratio. And that ends our discussion for the monohybrid cross. In the next video, we'll be talking about the dihybrid crosses.